We welcome everyone to the second talk of the day. We have with us Dr. Sivachandiran Somasundaram, and he is going to deliver a talk on synthetic biology and its application in health and industry. This session will be chaired by Dr. Ganesh. Dr. Ganesh is Assistant Professor of Research at AVMC. He completed his PhD in biotechnology and environmental engineering at the University of Ulsan, South Korea. He obtained his MPhil in biochemistry and molecular biology at Pondicherry University. He has worked as a postdoctoral researcher at Korean Research Institute of Biosciences and Biotechnology at Pohang University of Science and Technology for about 7 years in South Korea. His areas of interest include genetic engineering, microfluidics, on-chip PCR, synthetic biology for the production of isoflavonoids. His major work is on the synthetic circuits for diagnostics, and he has published more than 22 research articles. Thank you, sir, for joining us. Sir, we request you to chair the session.
Thank you, Dr. Adti, for your kind introduction. Dr. Adti, am I audible?
Yes, sir.
Thank you very much. I felt much delighted to introduce Dr. Sivachandiran here. So I have known Dr. Sivachandiran for the past 10 years. Dr. Sivachandiran Somasundaram is a postdoctoral researcher in Professor Mark Blenner Research Group, Department of Chemical and Biomolecular Engineering, University of Delaware. He did his PhD on bioengineering of microbes for the production of value-added chemicals at the University of Ulsan, South Korea. He works on engineering of Eroya lipolytica organelle for the production of and compartmentalization of oleochemicals and activation of cryptic pathways of this yeast using Crispr. So now I cordially and kindly invite Dr. Sivachandiran for his presentation. Thank you.
Thank you, Dr. Ganesh. Thanks everyone for joining the session. Okay, it's my honor to present here for today. And Dr. Ganesh is my lecturer from my undergrad. Just followed his path. So I did my PhD from Dr. Ganesh lab as well. I'm junior from him. So I'm going to present about synthetic biology for today. I'm going to talk about general what is synthetic biology, and synthetic biology has been growing for past 10 years. So I'm going to talk about general concept of synthetic biology and where it get applied. Yeah, I'm going to share my screen. Dr. Ganesh, can you see my screen?
Yeah, you're visible now. Yeah.
Okay, thank you. Okay, so as Dr. Ganesh introduced me, I'm from University of Delaware from Mark Blenner lab. So we've been doing so our lab is essentially focused on cellular engineering and applies in bio in different species, and try to develop genetic engineering tool that can apply to engineer the eukaryotic species. So let's see what is synthetic biology. So synthetic biology has very broad definition. The field has been growing only for past 10 years. It's been applied in many areas right now. So it basically means engineering life or engineering any species or organism. It essentially means reprogramming the biology for the application of human health, the application of pharma, application of biotech industries, and it's been widely applied. This field has been widely applied to make fuels, drug, natural product, and to make antibiotics and chemicals, food. It has vast potential in future that the synbio field can be applied in many areas that can largely impact humankind. So for the past decade, we know how the computer technology has been evolved. So similarly, there has been for past several decades. So we try to understand what is cell, how it works, how it functions. So just maybe in 1950s, we first know about genome. So we first know about what is DNA, and then just 20 years before, we completely sequenced the human genomes. And then just past like 1 year ago, we finished the task, like 100% like sequencing task completed. So we know. So now, like basically, a cell has been the like blueprint of a cell is essentially like coded by its genome. So what we try to do is that we try to like reprogram those codes. So like what happened if we like reprogram those code, and so we can like change its like phenotype and change its like behavior in terms that can be applied for the human health. So just like a small schematic diagram, like where are these symbiotic field can be applied right now? It's applied largely in like biomanufacturing, since we all know that climate change is real. It's no longer a fake news, and it's been like going so far. And the COP like 27, it's been, the summit has been happening in like Egypt now, and they try to like completely like reduce the dependence in the like fossil fuel. So we need to we need to replace this like chemical by different modes. That's why we use like microbes as a cell factory to make the same chemicals. So right now there has been like many like synbio work has been done to make different kind of chemicals. Like many like laboratory are trying to like scale up those for the fermentation level, and like possibly in like 5 to 10 years we can like largely see these chemicals like largely being produced from the microbes, like no longer from the fossil fuel. And the symbio also applied in the health and like therapeutics, like development of like Crispr to like diagnose a disease. For example, like recently the COVID. So some laboratories, like some symbio like laboratories, they have been developed based like sensing like method for the COVID-19 virus, and it has been applied also like in the plant system largely. Recently, like the last year, they designed, they like reprogrammed the root structure. They designed a gene circuit. In a way, they now can able to control like how the root can be grown, like whether the root can be grown like horizontally, or whether the like root can be grown as vertically. So they reprogrammed the system in a specific plant root. So the reason they did is that so they can like manage the plant root in a better way, that it can take up like nutrition from the soil, so it won't get a lot of water from the soil. So we can plant like engineered plant in a like desert area, or the like area which has like less water. And the mainly they also like use this field to mitigate like the climate change and like environmental change. Largely, like some labs, they've been doing to engineer the cyanobacteria. We know the cyanobacteria is the one that just use CO2 as like CO2 on like sunlight as like carbon source. It doesn't need any like glucose or any other substrate. So they try to engineer this cyanobacteria so that it can so we can produce like all this chemical like biofuel, lipids, like alchemical and natural product. And interestingly, they also start to use this symbio in the space biology. I mean, like we know, like humankind has been trying to like colonize the Mars. So in NASA, there is a unit called like AIMS Research Institute. They've been they've been like getting fund in many synthetic biology labs to try to engineer species like cyanobacteria to grow in the Mars. It's like a small like picture or like diagram how the Mars can be colonized in the future. Okay, I'm going to talk about like a few applications has been done in human health and to make like chemicals by using the synthetic biology. So the first study, like we're going to talk about like engineering the like microbiota. So we know, I mean, like human has been evolved along with like microbes. So in our body, we have a lot of the bacteria or like fungal or like virus, like human species or like any other like life form has been evolved together with with the microbes. So especially our like gut has a lot of the bacteria that provide like health benefit for the human body. And like recently we've been using a lot of antibiotics, and the global globally per year we use like 77 billion dose of the antibiotics. Like out of that, 62% we use like beta lactamase. So because of the overuse of antibiotics, we've been seeing a lot of drug resistant like antibiotics or like multi-drug resistant like antibiotics, especially like Clostridia. So what this group of the laboratory did is that they engineered the lactobacillus. So what they did is that so the beta lactamase resistant bacteria, it produced the beta lactamases. It resists the like antibiotics. So they discovered the biosynthetic pathway to make beta lactamase, and they put this biosynthetic pathway in the lactobacillus species. So like once they engineered this lactobacillus species, they supplied it in the human gut, and when they like tested, when they observed, so they supplied the antibiotics along with the engineered lactobacillus species. So when they they parallelly did like two study like antibiotics with the engineered lactobacillus, and the other group has only like just antibiotics. So when they give like just antibiotics, they observe like a lot of the good microbiota get destroyed. So it created imbalance in the gut, and it also leads to the secondary infection, and it causes like other diseases as well. But when they did the antibiotics along with the engineered lactobacillus, they haven't seen the death of the good microbiota in the stomach, because the antibiotics is not necessarily just kill the bad bacteria. It also, it also harmful for the same beta lactamase. It also causes the death of the good bacteria that present in the stomach. They studied the mouse model. They have seen just the ampicillin alone. They see the change in the colonization of the bacteria of the microbiota, and they see largely the growth of the Clostridium. When they supplied the antibiotics along with the engineered the lactobacillus, they don't see large growth of Clostridium, and they also observed the healthy amount of the gut bacteria as well. So the engineered the lactobacillus supplement in the gut, it really helps for the practice. The second topic, the second part of application I would like to cover is that the secondary metabolite production. So we know many of the drug for human health, we just usually get it from the plant or fungus, from their secondary metabolic the components. So this is just one of the example of a plant, what are the chemicals it can make. So we see alkaloid and amino acid, flavonoids. The flavonoids, it helps in for the antioxidant, and it does have effect on anti-cancer property. There's so many properties. So there is a group from UC Berkeley lab called the PA is Jake Kessling. This lab recently, 2 months ago, they produced anti-cancer drug. This is a secondary metabolite. The components, they usually obtained from a plant called Citronus rosea leaf. They used to get this drug from the leaf of the plant. So this is the drug has been used for many type of cancers, including lung cancer, brain cancer, like melanoma, testicular cancer. And to make just like 1 gram of this drug, they have to use like 500 kilograms of dried leaves to just make like 1 gram of the drug. And so expensive as well. And we know it takes like long time for a plant to seed it and like grow it and to extract it. I mean, it's a long bioprocessing time, and plant have a longer lifespan as well. So like recently, like the US agency, they listed this drug as like shortage drug in like 2019 and like 2020 because they cannot able to grow a lot of amount. So this group has been recently they like identified the whole biosynthetic pathway that present in this plant, and they put all this pathway in a yeast. This is the like 30 different step pathway. This is like a long pathway that present in the plant. So they took all these genes and the pathway like all together, and they put in the yeast. The reason is that yeast is the closest evolutionary like related species for plant. Both are like eukaryotic species. Like yeast has like subcellular organelles. Like yeast does have peroxisome, does have like mitochondria, but bacteria, like we know, the bacteria don't have any like cellular like compartments. Plant and yeast do have. So when plant makes like certain kind of like drugs, it not just made from the cytosolic space of the plant, because the plant cells also contain different like cellular like subcellular organelles. So it has to branch out at different like cellular compartments to make a certain drugs. That's why they choose the yeast, since it also have the subcellular compartments as well in the yeast. And they put all these like 30 different like biosynthetic pathway at like 3 different like modules. First, they try to make the streptozidin. Streptozidin is the very famous pathway, and like many groups has been trying to like produce the streptozidin in the yeast. So they designed all its pathway and put in the yeast. First, in the first module, they successfully produced the streptozidin, and they improved like 1,000-fold than the like native yeast. When they engineered the yeast, the streptozidin, the molecule, the fold has been increased to like 1,000. The titer was like 25.2 milligram per liter in a deep well plate. They haven't used the shake flask. They just use like a deep well plate that's very like minimum. And they introduced a second like module to make the molecule called like thrombocerin. And they introduced the third model, and they made like the vinolidin. From that, they like finally made the vinblastine. That's the anti-cancer drug. So in the final step, after they engineered, after they engineered the yeast using the synthetic biology tool, they got like 23.9 like microgram per liter of the vinblastine from the yeast. So now they're trying to like scale up this, and probably I would expect they will get succeed in like few years. So those are the 2 studies we just observed for the synbio application in the like mid drugs and the microbiota engineering. And the other topic I would like to cover on synthetic biology on making like alcohols, essentially like fatty alcohols. So the reason why we want to make like fatty alcohol, so the market for like fatty alcohol is expanding to like 7 USD like billion in 2050, because we need fatty alcohol to make all its product, including toothpaste, lubricant, like perfume, all of the stuff, like most of the stuff we need the fatty alcohols. And like primarily the fatty alcohol comes in like 3 different segments. One like short chain, another medium chain, and long chain. So the medium chain dominate in the industry because those medium chain fatty alcohols that comes from C11 to C14. That means like carbon, like 11 carbon to like 14 carbon chains that dominate in the industry, because from that they make soaps and detergent. The long chains, it has been like surging demand because of the growth of the cosmetic industry. It comes with from the carbon like 15 to carbon from carbon 22. These are the listed companies. They make the fatty alcohols right now. One from India too, like Godrej industry as well. How this companies they make this fatty alcohol? They use like two methods right now. One from the fossil fuel. They from the fossil fuel, they extract the crude, they refine it, they make like ethylene, and then they oligomerize, and then they make like fatty alcohol and its like derivatives. And the other method, they use the palm trees. So they grow the palm tree, they extract palm oil. From palm oil, they make this like all different chain lengths of fatty alcohol. And so right now, like 72% of the fatty alcohols are made from the palm trees, but it's still not like sustainable. The reason is that because of the impact, because the palm trees has been like largely grown in like Asian countries, especially like Indonesia, like Malaysia. These two countries shares like 75% of the palm export like around the globe. If you go to any supermarket, you find at least like 50% of all packed item that all contains the palm oils. I mean, all made from the palm oils. But there is like a huge effect from the palm trees, because they they destruct all the forest in the Indonesia, like Malaysia, and they essentially plant like huge amount of the palm farms that disturb the natural eco habitat, and it endangers the species, rainforest extraction, habitat loss. It also creates the social problems because it impacts the life of the indigenous people and the native peoples. So how can we go farmless future? So that's why we like to apply as a microbes. So here we use Synbio as a tool to make the same palm oil to make fatty alcohol. So we do we see microbe as like a cell factory, just like a chemical factory. We think like a microbe as a factory, so we can make the same palm oil from the microbes. So here we did some subcellular engineering. We choose a peroxisome. So a cell contains a peroxisome because it naturally had a beta oxidation cycle. So what it does is that any beta oxidation cycle, it recruit fatty acid, and it cleaves the chain lengths. For each cycle, it cleaves 2 molecules of the carbon and released as acyl CoA. And then you get different chain lengths of fatty acid. So what we tried is that we just localized a fatty acyl reductase, this enzyme that reduces the fatty acid to fatty alcohol. So we try to localize in the peroxisome, so to get fatty acid to fatty alcohols. And then we successfully engineered species called Aeroeoliprotica. So the reason we choose this species is that Aerovia, it naturally evolved towards high flux for the lipids. So to get fatty alcohol, we need large amount of lipid. This species naturally evolved to contain large amount of lipids. So it's very easy to make us the fatty alcohol from fatty acids. So we localized the GFP inside the peroxisome. This is a single cell of yeast. It's a peroxisome fused. So yeah, we got around 1 gram per liter of fatty alcohol. So another interesting topic I would like to I would like to cover the aspect of synthetic biology is that genome engineering. Let's see the history of genome engineering. So it's been a decade, and more than a decade, people have been trying to engineer the genome of a species. Any species, bacteria, yeast, or human cells, anything. So the past, people started to use meganuclease and zinc finger, nuclease and talon, and now is the era of the Crispr. So essentially, what it does is that it goes and cleaves. It breaks the DNA strand in the genome, and you can repair the those cleaved the template by inserting your targeted gene. and you can I mean, the advantage of using the CRISPR is that you can precisely you can double cut any genomic region in the whole cell. So after the discovery of the CRISPR in 2013 from Jennifer Doudna lab, the CRISPR application of CRISPR has been improved a lot in the research lab, in the research laboratory. You see the number of publications that just comes from the CRISPR. Yeah, from 2013 it's been improved a lot. Still, many CRISPR tool has been in the development. So let's see what's CRISPR. So CRISPR is essentially like a nuclease enzyme. It's like an enzyme that people they first identify from a bacteria. Some bacteria it naturally evolved to resist the virus. So when they try to study how it can resist the virus, they identify enzyme called the Cas9. So how this enzyme works? So what people now they try to do with any type of cell is that it comes with a two system. One is Cas9, another is the guide RNA with scaffold. So this is spacer RNA. So you can change the spacer RNA. It's green segment is a spacer RNA. You can change it to any type of sequence that complement towards the gene sequence that present in your genome. So based on that, it can go and bind to the target, the genome part of the cell, and once it bind, this guide RNA it form a scaffold, and then this will, the scaffold will recruit the Cas9 towards that region. So once it recruit the Cas9, this Cas9 it has a function to cleave double stranded of the DNA. So once it gets cleaved, the DNA get breaks, and our cell it naturally have the DNA repair mechanism, but unfortunately, it does non-homologous end joining. So if you don't have any template with homologous arm, it goes and repair itself, but sometimes it leaves a single base pair change, even a single base pair change that cause the collapse of the entire gene or the collapse of the entire cell. So that's why in many laboratory they used to have a complementary strand, so you will get homologous direct repair, so it won't have any marks when it gets repaired. And in 2019, US from NIH, they first used CRISPR to edit a patient's own immune cells, the T cells. What they did is that they engineered the T cell so the T cell can better see and kill the cancer cell. They engineered a receptor signal, and the engineered the T cell has been grown in vitro, and it get reintroduced to the patient, and it can fight the cancer. This is a timeline of cell and gene therapeutics. So as we discussed, to engineer the genome since 1917, people has been tried different methods to edit or knock out or knock in or repress or activate the gene in the genome of any type of cells. And the first gene therapy trial has been started in 1990, and they used different method to engineer the genome in a cell. So once we found the CRISPR around 2012, so CRISPR is essentially it's just act as a catcher. It catches any part of our gene in the genome, or any segment even. And then the first CRISPR trial for lung study has been happened, and they have like a long plan till like 2030. We call it as like Crispr era as well. And there has been a lot of especially from like MIT Broad Lab, like they've been doing a lot of like, CRISPR study for the human cells. Yeah, and I would like to cover like some of the epigenetic stuff as well. So CRISPR, so let's see what is like epigenetics. So it's like epigenetics is a study of the like heritable gene expression pattern that does not involve the changes in the DNA sequence. So far we know, like any type of cells in our body, like regardless of like skin cells or it's like it's like heart cells or like muscle cells or like any type of cells, it contains the same same codes of the DNA like sequences, like 3 billion, yeah. But each cell act like differently from like genotype and like phenotype. So why that different happen? The reason is because of the epigenetics. I mean, it's not just the code; it also important how the code get like read in the cells. So liver cell, it read the genome in different way than the hair cell, than the brain cell, than the skin cell. So based on the epigenetic marks that present on the on the DNA, the way it get like read and it like produce enzymes is also like different. So like DNA is not like free floating in our cell, like we see in the textbook or like movies. Like we know that like DNA is like densely like packed in like a chromatin like structure. It has like histone. That is the DNA segment. It's the DNA that like coiled like histone proteins. So this is histone. It's the DNA strand. This is like a tightly packed. So if like DNA is like naturally it's like a negatively charged, and it's like tightly packed, and we can if the DNA is like tightly packed like around the histone molecule, this part of the gene cannot be transcribed. The reason is that it's like tightly like packed in the histone. So the transcription, like the transcription initiation molecule, it cannot have the access to like bind this like DNA segment to read it, and accordingly it cannot make its like mRNA, cannot make its like protein. So so what people like now now they're trying is like they try to like reprogram these like epigenetic like modules. There are like many different like epigenetic marks that present in the cells. Like most important marks that present in the cells are like gene methylation and like histone acetylation and histone like methylation. So now with the help of CRISPR, they also like try to modify this like epigenetic marks that present in the cell. So like what they do is that like I showed you this like Cas9, it has the function to go on like it has the function to go on like bind in a like specific DNA segment and like cleave this like DNA. Here they engineered this Cas9 into dCas9. So like dCas9, like which means like it is not like catalytic, it is not catalytically active, but it goes and bind at the specific DNA site. They like knocked out like 2 protein that present in the Cas9 that has a function to like cleave the DNA strand. They like remove those 2 specific amino acids. Sorry, yeah, I'm gonna like finish it. So they knocked out protein, and then this Cas9 has been like used to specifically bind with. So along with the Cas9, they use like epigenetic like modifier. So this modifier can goes and like change the epigenetic marks present in the genomic region. So they can like, because like a lot of cancers are like the epigenetic problem, like more than like a problem from the genetic code. The codes are same, but the way it function, it differ in the cancer cells. the change happened because of the epigenetic mark that present in the cancerous cells. So that the way they try to control and change the marks that present in the cancerous cells to make the cells become to act like normal cells. Yeah, I would like to thank my lab, Professor Mark Brenner, and my funding agency NASA and NSF and my lab groups. Thank you.
Thank you, Dr. Sivachandiran, for elaborating the synthetic biology and its applications in various fields. You talked about the antibiotic resistance mechanism and also secondary metabolite production, especially vinblastine, as it is used as an cancer drugs. And also you talked about the subcellular engineering for the fatty alcohol production. And finally, you end up with the CRISPR-Cas9 and how is it used for the epigenetic markers. I have a few questions to you. For the fatty alcohol production, what tool you used?
Yeah, we use CRISPR. Sorry.
What are the bottlenecks you faced?
Yeah, the bottlenecks we faced is more the target integration. So it is not random integration. So generally people go for to engineer the eukaryotic species. They go for a random integration. They don't target at a specific locus chromosome. But here we use the CRISPR to target at a specific locus in the east chromosome. There we knocked out and we put our gene on the chromosome in the east. So it took a long time for us to make it happen. And once you knock in your gene in a specific locus in the chromosome, then the yeast become stable. The titer never change, more stable expression.
Okay, thank you very much. And one more question is it possible to detect any on-site kit to detect the biomarkers by using the CRISPR-Cas9?
I think the people has been developed for especially for COVID. I've seen that last year, and unlike many labs are trying to develop this diagnostic kit by using the CRISPR-Cas9 system, because it simplify the diagnostic methods. I mean, in future, I will see a lot that comes in CRISPR-Cas9 based diagnostic method.
Okay, thank you very much. Thank you giving for your valuable time, the late night. So I don't find any student questions. Thank you very much. Over to Aarti.
Thank you, sir, for a very interesting talk, the latest and upcoming research. It was very enlightening. Thank you very much. As a token of appreciation, we would like to honor you with a memento and a certificate.
Thank you. Thank you.
We would also like to honor our chairperson, Dr. Pranesh, with a memento and a certificate. Thank you very much, sir. Thank you for being a part of BMSCON. Thank you so much. We'll move to the next presentation.